Now that we've learned about amino acids and that they connect to each other to form a peptide bond, which is going to lead to larger protein structures, larger polypeptides, we're going to learn a bit more about the big picture of protein structure. And to begin with, right, let's look at a couple of different cases. We have enzymes, which we've looked at before. These are a type of protein. We've considered an enzyme we'll call maltase, a globular protein with an active site to bind the substrate maltose. Remember, that's a disaccharide that we're going to split into basically glucose units. This is kind of like a ball, right? You know, the protein has this ball globular shape and an active site that's going to connect with maltose. This is one type of protein, globular proteins. Hair and nails are made out of keratin. This is another protein, and this is a fibrous protein that has long fibers, they're strong, and this is from other chemical properties, right? Very different in its big picture molecular appearance compared to maltase. And so, you know, these pictures strive to show this. They don't show the individual polypeptides, but they do show the big picture of what the protein looks like. And again, these are shown in this way because proteins are often, you know, thousands, tens of thousands, even more molecular weight. And so we do need some kind of a shorthand. It's the diversity of the amino acid structure that allows for this tremendous variation in protein structure, and this variation in protein structure has huge variation in function. And I think this highlights that really well, you know, that we have basically a molecular factory on a globular protein as an enzyme. Not exactly a factory, but similar. Whereas we have basically molecular armor for keratin, right? Very, very different, yet built out of the same things. And so the first level of structure is the amino acid sequence, and this is a small protein, and it's actually made of two distinct chains. But it highlights this well because in the A chain of this protein, we see just a sequence of amino acids, and in the B chain, we have a sequence of amino acids. And notable here is these connectors with S is called a disulfide bridge. We'll talk about that soon. This from cysteine, which contains sulfur on its R group, is a way that polypeptide chains can be attracted to one another or even link among the chain. However, the primary structure of an amino acid is defined by the amino acid sequence. And to define this particular structure, we have one sequence for the A chain, and we have a different sequence for the B chain. That's the first level of structure, is just how the amino acids are connected. And there are 4 levels of structure. Secondary structure comes next. The resulting amino acids make up regional variations along the polypeptide. And for a polypeptide, we may have some that's alpha helix, then some that's beta pleated sheet, but these are the two motifs, right? We have an alpha helix where the polypeptide chain curls around and it makes kind of like a coil. A beta pleated sheet sits in kind of like corrugated cardboard, I suppose, and has interactions that hold it in that position. The alpha helix has interactions that hold it in its alpha helical position. These are regions along a polypeptide chain that result in these local variations. So how does this occur, right, from the amino acids, right? We've talked about here that there's this coil structure and that there's this corrugated cardboard beta pleated sheet structure, but how do the amino acids determine this? And so here's an example where we have kind of just a straight polypeptide backbone showing interactions between parts on this backbone that we may see on a protein chain. And so 4 examples of what we might see. Right, we can have one R group make an ionic bond with another. That would certainly cause interaction, right? If we have a negatively charged R group that can see a positively charged R group, they're going to see each other and they're going to pull the polypeptide chain together in that way. Hydrophobic interactions, right? We know this idea of like dissolves like, and polypeptides are usually going to be in some sort of an aqueous environment that is going to push hydrophobic stuff together and pull hydrophilic stuff towards the water. This can cause an interaction between those sites on the polypeptide chain. We saw this disulfide linkage previously, and this is a really important one because this makes a covalent bond, which is stronger than these other classes of interactions. And we can see that only on this one specific amino acid that contains the sulfur cysteine that we saw previously. This can actually create a covalent electron sharing bond between points on the polypeptide chain. And finally, hydrogen bonding exists as well. This is the force that attracts water to itself, and these are all things that we've seen before, right? So this is that diversity, right? We have huge diversity in amino acid structure, 20 different amino acid R groups, and proteins make their structural diversity based on the fundamentals of molecular interaction that the R groups possess. So with that in mind, right, the overall protein structure, we have amino acid, that's our primary structure, the amino acid sequence. Secondary structure, alpha helix versus beta pleated sheet. The tertiary structure is going to be the overall shape of just one polypeptide chain, but proteins do in fact often have multiple polypeptide chains. So hemoglobin, for example, has a heme group plus 4 of our different polypeptide chains, right? So it's more complicated than just a single polypeptide chain. It can be multiple chains coming together to form a larger functional protein. That's the quaternary structure.